Okay, this lecture is on viruses. What we're going to look at in this lecture is how viruses differ from cells. So viruses are non-living. We're going to look at how viruses are classified based on essentially their makeup. We're going to see how we can also culture viruses in the same way we culture other microorganisms with minor modifications. And we're going to look at the replication cycle of a couple different viruses. Okay, so viruses are essentially ubiquitous. So we've heard this word before referencing bacteria, that bacteria are ubiquitous. They're on our body, within our gastrointestinal tract, within the environment around us, from the atmosphere to the oceans to the terrestrial environments. But actually, viruses are even more numerous than bacteria, therefore making them the most ubiquitous. So they outnumber bacteria about 100 to 1. Looking at viruses, viruses can cause human disease. So this is mostly the reference we look at for viruses. And at the end of the lecture, we're going to see some viruses and how they can cause human disease with their infection cycle. Looking at viruses that infect bacteria cells, we call those bacteriophage. A bacteriophage is just a specific name for viruses that are working with bacterial cells. So bacteriophage do not infect our cells. But they can be routinely used as cloning vectors, meaning that we can use this as a way to genetically modify other bacteria. So this can be used for cloning and gene technology. We can also grow bacteriophage in the laboratory, and that's what we see on the bottom right image. So the opaque nature of the plate that we see, that's actually all bacteria. That's a confluent lawn of bacteria growing on the plate. Those little holes that you see, those translucent holes, are actually areas where there is no more bacteria because the bacteriophage itself has gone in and lysed open the cells, creating a clearing around it. We call this clearing a plaque, as seen. So we see plaque on the image here, and it's pointing to those clearings, those holes. Everywhere we see a hole on this plate is a place where a bacteriophage landed, entered into a cell, replicated, and lysed open the cell. Then we released 20 to hundreds of viruses around them. Those viruses infect the surrounding cells, go in, replicate, lyse open those cells, and then we have another release layer of viruses affecting the next cells around them. And this continues on until we see a visible clearing, a plaque on the plate. We can numerate viruses based off of what we call a plaque assay. So every hole in the plate that we see here represents one virus that landed on the plate and broke into that cell and then surrounding cells, creating that clearing. Similar to the way that we enumerate bacteria based off of colony production. Okay. So this is a research abstract from a professor at UCI, Luis P. Villarreal. And I want to read it because I think it gives a broader understanding of virology itself. He's a virology professor and researcher. So here he says, in the last decade, metagenomic assessments have led us to realize that viruses are the dominant biological entities of the biosphere and are the most numerous, diverse, and dynamic genetic agents on Earth. Let's go ahead and just break down that full first sentence because that's really a mouthful. So here he says viruses are the most dominant biological entities of the biosphere. So they're the most dominant, not only because they're numerous, but for many other reasons as well. It's because they're essentially on top of what we would see as a food chain. Viruses can infect all forms of life. So we have viruses that affect eukaryotic cells, viruses that affect prokaryotic cells. Viruses are essentially on top of that food chain. So the most dominant biological entities, not only by number, by being diverse and able to affect all cells around it. They're also dynamic genetic agents on Earth. Because we already talked about the process of transduction, of using a virus to genetically modify a cell. So we can alter genetic information within a cell by the use of a virus. So moving on, it says, although viruses have long been dismissed from the tree of life, a simple, destructive, and selfish extragenomic genetic parasite, comparative genomics now makes it clear that viral colonization distinguishes all domains of life. It says now he's tracing how viruses have contributed to host group survival from bacteria to human social evolution. So looking at viruses, we don't put them on the tree of life because they're considered non-living, but viruses are actually exchanging DNA on the tree of life. They have horizontal transfer of genetic information, and we have retroviruses that can insert their DNA into our cells, into our genome, integrate into our DNA, so that every time our cell divides, we replicate the genetic information of the virus itself. So we can see that actually the tree of life is littered with viruses, whether they be selfish or whether they be beneficial. This is quite an old research abstract, but when we look back at the study of microbiology and start with bacteria, we typically start with bacteria that are pathogenic, the ones that are causing disease. Following that, we start understanding how important our relationship is with our own microbiome or microbiota within our body, within our gastrointestinal tract, to train our immune system to produce vitamins, to fight off pathogenic bacteria. Well, we're going to see probably the same thing with viruses here, that not all of them are bad, but of course, the initial study of virology is really bringing up the more parasitic viruses, the harmful virulent viruses, than the ones that are actually benefiting us. But even if we look at the process of pregnancy within women, the uterine lining, actually, the DNA that codes for the uterine lining is actually a remnant of viral DNA that remains. So we see that our species actually had a boost from an infection with a virus that made our survival of our offspring higher. So when it comes to evolution, we see viruses as being shaped our evolution physically in allowing the production of offspring to be more beneficial for us. And here we can see it says host group survival from bacteria to human social evolution. So I think social evolution is another element that we don't really think about. So I really like this research abstract because it makes us think out of the box about viruses. We're not only thinking about COVID, we're thinking the greater implications of virology. Okay, looking at virus replication within a host cell, here we can see a bacteriophage. So this is a transmission electron microscopy. As it looks almost like an X-ray, we're seeing through to the inside of the cell. All of these shapes that you see here, this is the head of a bacteriophage. So they've infected the cell, and within the cell, they're essentially reproducing with the machinery of the bacterial cell. The protege here, we call the viron. So these are the viron particles that we see replicating within the host cell. Looking at different types of host cells, we know that we have bacteria with bacteriophage, but all forms of life can be infected by viruses. And this is why they're so important for crafting the tree of life, for shaping it essentially. So here we see plants can be affected by viruses. We can have large host range or small host range. Within plant species. Here's the tobacco mosaic virus. Tobacco mosaic virus was one of the first viruses to really be understood and identified. And we know because it was affecting our crops, tobacco crops. So it was studied in quite a bit of detail as we were having an economic impact by loss of crops. So the tobacco mosaic virus, TMV, can actually infect over 350 different species of plants and causes this phenotypic discoloration that we see here on the right. Okay, let's go ahead and look at measles. So we've heard of measles before. Measles host cells are human cells. So here's our globular eukaryotic cells that we have. And here we actually see the virus, the measles virus. So this is a transmission electron microscopy image. and we see the virus budding off from the cell, we actually probably notice that the virus looks quite similar to our cell. And that's because the virus is actually taking its outer envelope from the membrane of our cells. So as it buds out of our cells, it takes part of the envelope of that cell with it. So it's derived from the host cell's plasma membrane. So it is globular looking, just like our cells, and it's really just genetic information inside of that membrane, as we see here. Okay. Looking at viral genomes, what's unique to them is that they can actually be DNA-based or RNA-based. They can also be double-stranded or they can be single-stranded. All forms of life are based off of DNA as the genetic material, not RNA. So viruses are non-living, and we see some interesting exceptions here. We also know that all life has double-stranded DNA, so viruses being non-living can be double or single-stranded. We also can have capsids around the viral genome. And some viruses have envelopes as well. Okay. What we see here is called the Baltimore model. This is how we classify viruses, and it's based off of the physiology of the virus and the components. So these groups that we see here, we can see DNA, DNA, RNA, RNA, are showing us what the genetic material is. So whether it's DNA or RNA. Not only is it what the genetic material is, it's whether it's double-stranded, DS for double-stranded, or whether it's SS, single-stranded, as we see here. On top of that, you may have caught eye of these pluses and minuses here. These pluses and minuses are actually telling us whether these are sense, the sense strand, or if we're looking at sense template strands, whether this is a coding or non-coding strand is essentially what we're looking at here. Okay. Here we can see some viruses we've probably heard of, HSV, the herpes simplex virus, parvovirus, coronaviruses, influenza viruses, retroviruses. So we have quite a few different viruses in here. Some things to point out are some unique aspects of viruses. Here we see reverse transcriptase. This is actually an enzyme that is inside the virus itself. The virus carries with it. So in most viruses, they don't carry their own enzymes. They don't have a metabolism. They're non-living. But reverse transcriptase is a unique enzyme that viruses can use. They can take their RNA and make DNA out of it. When we think of the central dogma, we go DNA, RNA, protein, and it's a linear process. But viruses can actually take transcription backwards, and that's why it's called reverse transcription. Reverse transcriptase, ASE, as it's an enzyme. We go from RNA to DNA. The reason why a virus might want to go from RNA to DNA is if it's going to incorporate its genetic material into the host cell. So if it comes in with RNA, we can't incorporate RNA. We can't integrate that into the host genome. We have to convert it back to DNA with reverse transcriptase in order to incorporate that DNA into our genome. Otherwise, the virus is dependent on the cell's enzyme. So for transcription and translation, it uses host cell machinery. So I wanted to point out some unique things here, like retroviruses in particular, come in with their RNA and then go back to DNA. Okay. Looking at viral genome sizes, if we're looking at RNA-based viruses versus DNA-based viruses, RNA-based viruses are relatively small. And these are what we call retroviruses because that RNA. DNA viruses, on the other hand, again, are quite large. So we could see this is the genome of HSV, the herpes simplex virus. So we have quite a few genes here. Okay. Looking at viruses replicating in cells, again, they're non-living because they cannot replicate on their own. They need a host cell, whether that be bacterial or whether that be our cells or plant cells to replicate in. Here we see a virus being engulfed by a cell, uncoding itself. We see its viral genetic information within the cell. So viruses are actually using the host cell machinery to go from DNA to RNA to protein to run the central dogma to generate proteins so that it can generate more viruses. Now, this unique pathogenic life cycle of viruses is really interesting because it can also be used for medical therapy. If we want to insert a gene in the genome, if we want to genetically modify, we could use the life cycle of a virus to genetically modify a cell. We could remove any virulent pathogenic genes within a virus and add in a gene of interest that we want. By adding in that gene of interest to the virus will carry as a vector that gene into the host, and it can actually incorporate that gene into the genome of the host cell. So this would be genetic modification. This is gene therapy, the example that we're talking about here. Now, because viruses are not independent and they rely on the enzymes within the cell, the host cell, it makes it harder to target to target viruses that are pathogens because we can't target their main enzymes. If we target their enzymes, we're actually targeting our enzymes. One enzyme that's unique again, though, is reverse transcriptase. Because our cells don't have this, it's an enzyme we can target for viruses. But there's very few viral targets to attack because they're so similar, because they're using so much of the host cell machinery that we can't really separate them from the host cell machinery, so without affecting our cells. Antiviral agents, so there's not as many of them, also can have pretty severe side effects. And again, that's because it could be altering the host cell machinery as well. And viruses can mutate very, very fast. So we know we have antibiotic resistance. We can also have viral resistance, especially in the ones that have very, very small genomes. Okay, let's go ahead and dive into viral structure. We have icosahedral shapes, which is one of the most common shapes that we see and has radial symmetry. It's actually made out of 20 identical triangles. So what's unique to this is that we don't need many genes to make these triangles. We can have one DNA sequence or one RNA sequence that codes for one triangle. And we can just keep using that code to make 20 of those triangles in order to make one viron. So you just make multiple triangles. So having this symmetry, having this icosahedral shape is extremely simple and efficient for viruses to keep their genetic material low while still having this complex structure that protects their genetic material. Okay. Some viruses have envelopes on them. So here we can see a transmission electron microscopy image of the virus inside an envelope here. The envelope itself can have actually spike proteins on it. So these are things that our immune system can pick up on or the way they interact with cells around them. Okay. We have many different shapes and structures of viruses themselves. We have filamentous, or what we call helical viruses, as seen here. These are actually built all out of the same protein. So even though they look like small worms, these viruses, this is actually a linear representation of them. And if you were to take them, they're almost like a helical staircase. And if you were to see each step of the helical staircase, they're just on repeat all the way up. So again, we can have that built-in redundancy. We can have a small genome, and we just take that one gene and keep making multiples of it in order to build the structure of the virus. Good example of a helical virus or filamentous virus would be the tobacco mosaic virus or Ebola virus. Looking at bacteriophage, again, the viruses that are unique to bacteria, they have this complex structure where we have the icosahedral head, but we have this entire tail region here. This tail region actually acts like a syringe to inject DNA into cells. So here we see the bacteriophage T4. This is one that infects E. coli, very common virus used in the laboratory. We can see that once it attaches to the cell surface, it actually compresses that tail. When it compresses the tail, it injects its genetic material into the bacterial cell itself, into the host cell. We also have amorphous viruses. So these are ones with no shape. They can change their shape based on the environment around them. So here we see an envelope and a core on this one, but there's not necessarily symmetry to this. Again, it can change based off of the environment that it's in. It can have viral envelopes with spike proteins. So these would be those spike proteins associated with it. Vaccinovirus and smallpox virus are these amorphous type viruses that we see here. This is a pseudo-colored image of a transmission electron microscopy image. So the core here, this is where our genetic material is going to be inside of there. Okay. So not only do we have viruses to consider as pathogens, we can also look at virons or prions as well. So viroids themselves are just actually RNA. So if we look at this image on the right and we look at this sequence, we know it's not DNA because we see uracil. But we see this unique structure folded back on itself. So it is double-stranded at times, single-stranded at other times, and it takes on this very, very unique structure. Typically, we think of RNA as mRNA, tRNA, and so on. But here we can see that viroids can actually be pathogenic RNA molecules. So they don't have a coat around them or anything. There's no protective capsid. And most of the time, our viroids are affecting plants. So it's genetic material, again, altering the genetic material of other organisms. Prions, on the other hand, is something you may have heard about before. So prions are different in their molecular structure because they're not made out of nucleic acid like RNA. They're actually made out of protein. So this is an amino acid structure. We can actually see the alpha helixes here and beta pleated sheets here. Now, prions themselves can interact because they're not necessarily coding in the sense that RNA is. They're a little bit different with this amino acid structure. It's more about their conformation or their shape and how they interact with other proteins. So here we see a normal conformation, the normal fold of a protein. And here we see an abnormal fold of the protein. So what can happen is that one prion can interact with another prion and misfold the other prion. So when we have proteins themselves, they can be held with primary, secondary, tertiary, or quaternary bonding. And these are all chemical bonds like ionic bonds, covalent bonds, disulfide bonds, hydrogen bonds that hold them together. So by altering the bonding pattern, we alter the conformation or the shape of the protein itself. Now, when we alter the conformation, we denature proteins, they can accumulate. And there's many neurological disease causes by prion disease because they can accumulate in the brain and actually be a physical blockage of signal to move through. A couple good examples of prion diseases that you may have heard about are mad cow disease, Creutzfeldt-Jakob disease, which is again can be consumed in meat. It would be affecting neurologically the cows, and it would affect humans the same way. So we see the host audience would not only be animals such as cows, but would also be humans in this case. We also have Scrippi and Kuru. Kuru is an example of a prion disease that can be passed from human to human by the ingestion of neurological tissue, such as the brain. So again, these can accumulate in the brain, prions. So if someone was to consume the brain of someone else, they could consume these prions, which would then go on to misfold other proteins within the brain, causing the accumulation and the death of this person. In some cultures, cultures are quite different from one another, but in some cultures, there is the concept of family members living through one another by the consumption of the neurological tissue, by the consumption of the brain of their deceased relatives. So by consuming the brain, that relative lives on within them. And so we see Kuru can be passed from generation to generation. So when the affected tissue is consumed, it causes Kuru, this prion accumulation within the brain, which causes an earlier death of the person, and then their relatives would consume them as well. So we can see it can spread throughout a population like this. Okay. Let's look at viral diversity and evolution. So we talked about bacterial diversity and genetic change. Well, we have the same situation of evolution affecting viruses. They are influenced by the environment around them, or they can be the environment that's influencing the bacteria or humans to be modified as well and change and evolve. So natural selection plays on them as well. If we look at genetic code, here we see a change from C to A. nucleotide sequence is changed. And once it's changed, we're altering that sequence, meaning we're altering the phenotype of the virus from genotype to phenotype. So natural selection may select for some of these interesting mutations. Again, with these small genomes, they mutate quite quickly, and we can get what we call antigenetic drift happening. So rapid mutations here with natural selection playing into that. When we think about every year, in the vaccines that exist for the flu vaccine, and it changes every year. We don't get the same vaccine for the same flu every year. That's because the influenza virus is always mutating and changing. So if we have the vaccine from one year, it might not be effective the next year. So the flu vaccine has to be constantly altered every year based off of new strains that come about from this antigenetic drift and natural selection on viruses. Looking at viral diversity and evolution, we can actually build these family trees with viruses as well. So even though they're non-living, their genetic material traces them back to a root common ancestor. So we have the tree of life where we can put humans and all other living organisms, including prokaryotic, eukaryotic cells. And then we can start looking at viruses as well, whether they're DNA, whether they're RNA based. We can see how they're related to one another. So we have herpes 6, herpes 7. We can see Epstein-Barr and the equine, horse herpes can be associated with one another. So again, it's based off of their genetic material. And the closer they are to one another, the more closely they are related. So alpha to gamma are more further related than these viruses within the gamma herpes virus family. But these are all herpes viruses. So these are kind of our zoster viruses that we see here. Okay. Looking at viral structure itself in influenza, one of the really unique things that we see with viruses is this exchange of genetic material. We talked about humans having diversity because we have DNA from the mother, DNA from the father that recombines, right? That's when the sperm and the egg come together. We said bacteria don't have that, but they have horizontal gene transfer where they can exchange genetic information. Well, actually, we have reassortment with viruses as well. What can happen here is that two independent viruses, two different strains, can actually infect one organism. So here we see an avian virus, so a bird flu, and we see a human virus over here. Both of them moving into the host, a swine, and we get this interesting swine flu, which is a combination of the avian and human virus. So if they both infect the same cell, they decode and their genetic material is readily available. When we start packing up new viruses, we can pull some of the DNA from the human virus, some from the avian virus, and all of a sudden we get this new reassortment of a new virus. And this is antigenetic shift. We see the red and the blue combined. And this can go back into the human population. So this is interspecies reassortment, as we're looking at 3 different species here. And this can lead to some of the most virulent pandemics that we are yet to see. As you may imagine, this is very common in animal agriculture. The closer we are to animals and interacting with them, the more likely we are to pick up these interspecies reassorted viruses that can be extremely dangerous. These viruses, because they are so new, these would be novel viruses, are not something that we would have a vaccine readily available for because it's something we've never seen before. It's novel. Okay. So we can have viral diversity on many different levels. Evolution will play on multiple levels. And let's go ahead and look at these different levels. So we can have viral diversity within a host community. So here we can see the host community can be different species. So just like we saw swine, just like we saw the avian host and the human host, we can have evolution between different species here. Then we can have it within a species population. So we can have different strains that start evolving within species populations and even within an individual organism, meaning just you yourself as an individual can cause natural selection on the virus, whether that be due to your immune system, whether that be due to antiviral therapeutic agents. The virus can change within your body. So as you take antivirals, natural selection occurs. The viruses that are randomly mutating and can evade these antivirals or evade your immune system will be the ones that survive and pass down. So we see evolution within a host community, within a species population, and even down to the individual. We see natural selection and adaptation occurring. Okay, let's look at viral replication stages. So step one, the virus has to interact with the host cell. So in order to interact with the host cell, we have to have recognition. This can be from spike proteins on the outside of the virus or some kind of interaction. The virus has to maybe trick the cell, or the virus has to attach to the cell in order to mount an infection. So it has to contact and adhere to the cell. Following that, the genetic information of the virus has to enter into the cell. So that is critical. If the viral information cannot enter into the cell, we cannot mount an infection. So the genome can enter into the cell a couple different ways. We see some viruses are engulfed by the cell and taken in. Others, like a bacteriophage, are more like a syringe where it's being injected directly into the cell. Following that, the virus has to start assembling to make its prodigy virons. So those are the essentially offspring of the virus. So we have to start making proteins and generating and assembling new viral particles. Then it has to leave the cell. We have two ways of leaving the cell. We can burst open the cell and release viral particles, or we can have budding. And budding is what we saw with the measles virus. Let's go back and look at that measles image really quick. Here we see budding out. So that membrane is again fluid and the virus can just bud out of it. It can just bubble off essentially, kind of like a soap bubble bubbling off. Okay, so we have a lysis or budding. So these are again the four general steps that we look at for viral replication stages. Let's go ahead and look at bacteriophage replication. So we'll get a little bit more specific with this one. And we have two really interesting phases to understand. We have a lytic phase and a lysogeny phase. Typical viral infection that we think of is probably the lytic cycle. So here we see a bacterial cell. This is its host genome. Here is the virus. It injects its DNA into the host cell. It starts making prodigy viruses, assembles them, and bursts out of the cell, lyses out. And the cycle repeats. This virus can go to a new bacterial cell and mount an infection again. If we're looking at virulent phage, we're going to look at the lytic cycle only. So this is virulent because it kills the cell. But we also have temperate phage. And temperate is a relationship between this virulence, lytic, and lysogenic cycle. So when we undergo lysogeny here, we can understand a slightly different cycle. So we start the same way. Virus injects its DNA. But instead of this viral DNA starting to assemble viruses and using the host cell machinery, what actually ends up happening is that the virus incorporates itself into the host genome. So it has integrase, and not all viruses can do this, but integrates into the host cell genome. So we see the viral DNA in purple, and in bluish white is the host genome. We see them incorporated into one another right here. As this cell divides, every time it replicates by binary fission, it divides into two, it's going to replicate its own genome. When it replicates its own genome, it in turn replicates the genome of the virus. So this is how the virus becomes a part of the bacterial cell. And this is for humans as well. If the DNA of the virus is incorporated into our genome, as we see here in this lysogenic cycle, every time our cells divide, the viral DNA divides with our cells. So that's what we see here. All the prodigy cells will have that viral DNA incorporated in them. These cells are now genetically modified with viral DNA. And you could imagine if this cycle happens over and over and over again, we would start accumulating viral DNA in us, which we have. Okay, let's go ahead and go back to this leg. If we're looking at the lysogenic cycle, so not only are these cells dividing and passing down this DNA, we're going to see that we can actually pop back out and merge back into the lytic cycle. So we can just be straight lytic, or we can be temperate phage and move between lytic and lysogenic. So the virus can incorporate its genetic information. So here the virus is only existing in nucleic acid formation in the form of DNA, of the A, U, C, and T. It's not the virulent form that we think of in the bacteriophage that we see here typically. So here it's still existing, it's still there, and it can pop back out. So it can excise itself out, as we see here, separated from the host genome. It can start making viral particles and become virulent and lyse open those cells or bud out of cells again. So why this is important, if we were to look at viruses with humans, we know some viruses can be latent, meaning they're not actively being expressed. So that's our temperate, kind of like a temperate phage. Our viruses can incorporate themselves into our genome and just lie latent, essentially, in this lysogenic cycle where they're not causing an issue. So herpes simplex virus is a great example of this, whether it's oral or whether it's a genital infection. They're at the root ganglia of the nervous system here is where the cells are actually infected. And similar to what we see here about integrating into the host genome, we're taking on this genetic information every time our cell divides. That genetic information divides with the cell. It's replicated with the cell. So this is why we cannot get rid of a herpes simplex virus infection. That DNA is incorporated into our DNA. That's why there's no cure. There's suppressive therapies that people can take that help prevent the virus from being expressed. And what that's doing is suppressing the virus, is keeping it in the DNA instead of having it burst out of the DNA, make viral particles that are then harming the surrounding tissues. If we're lysing open cells, if we're budding out of cells, these cells become sickly and we get damage to these host tissue. HIV is like this as well, that HIV integrates into the genome. So we can't just get rid of HIV once there's an infection. While we have plenty of new retroviral therapy, the retroviral therapy is keeping that viral DNA incorporated into the host cell's DNA. So it's not really harming us if it's in our DNA, but again, we can't get rid of it unless we go in and we excise out and cut out that piece of DNA. Maybe we use something like CRISPR to do that. That DNA will remain with us for the rest of our life. Okay. We talked about bacterial growth before. Let's go ahead and look at bacteriophage growth. So it looks similar, this graph that we see. Time on the x-axis, on the y-axis, we see phage, so it's a bacteriophage, the number of viral particles per milliliter that we're looking at. We have two phases that we break this up within. So it's a biphase kind of graph. We have the eclipse period, and then we have the rise period. So in the eclipse period, we see it go up and then slightly down, so just a small number. So what this is the virus actually initially getting into the body or into the system. Actually, here we're looking at bacteria. So the virus getting into the system, once the viral DNA is inside the host cell, before it starts replicating, we all of a sudden detect less virus, so it drops down. Right here is where the virus is all of a sudden starting to replicate. So it's producing proteins, it's using maybe the RNA polymerase and DNA polymerase of the host cell and assembling the viral particles, those virons. And then when we start this exponential growth phase here, the rise period, essentially, the rise period is when we all of a sudden start releasing viruses. And then we level off when we see complete cell lysis. So all of the viral particles, like we see here, have lysed out of the cell. So first we start increasing in viral particles and then level off, hit a stationary phase as soon as cell lysis is complete. Okay. Let's go ahead and look at what we've learned before about horizontal gene transfer. So these are some questions, discussion questions to talk about and think about with yourself or with others. Number one, so it's a throwback to the previous lecture, we see what are the three forms of horizontal transfer of genetic information? At this point in the lecture, I would pause and answer this question yourself. Take notes on it. Go back and look at your notes. Okay. So once you resume the video, we can look at question number 2, which is what are the two methods of horizontal transfer of genetic information that involve a bacteriophage? Again, I would pause here and look back at your notes. Okay, well, go ahead and start answering these questions now that you've taken your notes. Number one, the three forms of horizontal transfer of genetic material. So we go with the three basic ones. So we have transformation, conjugation, and transduction are the three forms of horizontal gene transfer. We also talk about jumping genes and transposons, but those are the three main ones, the conjugation, transformation, and transduction that we want to focus on right now. Now, when we get into the second question, what are the two methods of horizontal transfer of genetic information that involve the bacteriophage? Well, conjugation, we said, is cell-to-cell contact. We know that from our last lecture on that question. Or we have a sex pilus and we have the F prime and F plasmid and HFR. Following that, we can look at transformation, which is where cells are picking up exogenous genetic information around them, which we saw with the mouse experiment, which is a natural form of transformation. They can also be done in a lab. Then our last one that we talked about is going to be our transduction. Transduction involves a bacteriophage. Bacteriophage, again, is the virus that infects bacteria. And we have two different ways. We have specialized and general forms of transduction. So those are the two methods of horizontal gene transfer to transferring genetic information with a bacteriophage. Okay, let's go ahead and move on. Let's talk about culturing bacteriophage, how we can get them to replicate. So we already saw a picture of plaques, but let's go ahead and see how we would actually do a plaque assay. A plaque assay is the title of the experiment we can do to culture a virus and numerate viruses as well. So what we do here is we have phage, just like we would have bacteria in a culture, and we would actually combine it with another culture of bacteria. So viral culture plus bacterial culture together. Because remember, viruses cannot live on their own. You cannot just plate them. You have to plate them alongside their host cells. What happens in this mixed culture of bacteriophage and E. coli is that the bacteriophage start infecting the E. coli. So that's a microscopic look of what would be going on. From there, we're going to add this to what we call molten agar. Agar is what is the medium, that nutrient-rich medium that bacteria grow on. So molten just means that it's liquid formation. So we actually pour that culture into the liquid agar. When we mix them together, we do what we call a pour plate. We pour it out onto an agar plate. What we see here is that we're spreading it evenly. And what we end up with after we incubate it here, it says 37 degrees overnight. That's body temperature, 37 degrees Celsius overnight. What we see is that we end up with a bacterial lawn. All of this that we see here is all bacteria, a confluent lawn across the plate, except it has the holes in it. And again, those are the plaques here. So those holes that are drilled out from the virus going and infecting a cell, replicating, lysing out, and infecting the surrounding cells. Then again, those surrounding cells producing viruses, then bursting out, and then affecting the surrounding cells. So it's like a hole that just gradually gets larger and larger until we can actually visualize it. As our plates are about this big, we're actually able to enumerate viruses as one plaque represents one virus that landed there. Okay. Let's go ahead and look at a couple different viruses. We're going to look at some unique to humans like HPV, the human papilloma virus. We see that it's infected about 80% of adults. These facts actually may be a little bit older because now we actually have Gardasil. We have a vaccine to protect us against this virus, but this vaccine only works pre-infection. One of the concerns with HPV, the human papilloma virus, is that it can cause cervical cancer. Cervical cancer, that's hyperproliferation of the cells within the cervix, causing atypia of these cells. So this is typically what pap smears are for, is to look for any abnormalities or cervical cancer potentially caused by HPV. So this is a good association, how we can see viruses can cause cancer. It's not just our own genome and our genes. This is essentially a contagious form of a cancer. Okay, HPV is highly infectious. We can see that with 80% of adults being infected. It's transferred through sexual contact. Genitally, also orally. You can get an HPV infection within your throat, which can lead to throat cancer as well. The best way to protect yourself from HPV infections is protection. That would be condoms. Condoms don't always provide 100% protection, though, from HPV, as HPV is not only found on the genitals, but around the genital regions, and condoms don't cover all of these areas. So they don't protect 100% against HPV. So while this is a barrier method, it doesn't block all contact in this case. And again, oral exposure can also pass HPV from person to person. Gardasil is our best protection that we have today against the viruses. It works against multiple strains of HPV, specifically the ones that cause cervical cancer is what we're really looking at. But the Gardasil vaccine has to be given around the ages of 11 to 12 before sexual contact is ever established. So before an age of maturity with sexual contact, or else it doesn't work. If you already have an HPV infection, this vaccine will not help. Okay, let's look at the papilloma virus here. So we can look at the genome of it. Again, these viruses have small genomes. It is DNA, so it's a little bit larger, double-stranded DNA, as we see here for HPV. And something unique to it, though, the way viruses simplify themselves is they actually have overlapping parts of their genes, so reading frames. So if we're looking at this here, this gene is overlapping with this gene. So part of the E2 gene is also the E4 gene. So instead of having the E4 gene as an additional sequence, as having more than 8,000 base pairs, it just is wrapped up within another gene. So they have overlapping reading frames. So reading from here to here is different than reading from here to here. And here we can see that's a human papilloma virus. And it only has about 8 genes. Let's look at how the human papilloma virus causes infection within our human cells or mammalian cells. We'll be looking at the replication cycle here. Starting with step 1, here we see the virus. So that is representing this virus that we see here. And it's actually interacting with receptors on the outside of our cells. These receptors are not made to receive HPV, but the virus is taking advantage of these receptors and being able to interact with them. So we have this cell surface interaction, followed by endocytosis that essentially tricks the cell to take in the virus, and it creates what we call an endosome as it's inside the cell. Now we have to start unraveling the virus. We have to get to that genetic material because that's the core of the virus itself. So the endosome bursts open, and we can see the virus starts breaking apart, and the genetic material found on the inside makes its way into the nucleus. So that's the genome we're seeing here. Those 8 genes only, simply making its way into the nucleus of the cell. So it tricks the cell going into the nucleus as well. The genome completely is uncoded and available for use, and we start DNA replication of the viral DNA. So here we go from 1 genome to 2. Not only are we replicating the viral genome of HPV, we're going to start also making the protein. So we're taking the DNA that's found on these 8 genes and making proteins. So we're running the central dogma from DNA to RNA in transcription and RNA to protein in the process of translation. So here we see we have our host RNA polymerase. We're making RNA here. We're making DNA here. And we actually see that the RNA ends up being protein. So this is a process of transcription. Translation happens in the cytosol. And these proteins make their way back into the nucleus to start assembling new viruses. So we go DNA plus protein. Then they can merge out of the cells. So they actually shed essentially out. The shedding of the epithelial cells is going to release the virus. So as our cells in our body are shedding, we have the virus shedding as well. We've heard of the term viral shedding. So this is why skin-to-skin contact with HPV can cause an infection. Okay. Another thing to point out here, when we talk about cancer development with HPV, the pathway is slightly different here. We have HPV can actually integrate into the genome. So once we have an HPV infection, if the DNA of HPV is integrated into our host cell DNA, we cannot remove it. So every time this cell replicates, it passes down that DNA to the next cell. So it's not that this cell could die off and become sickly or taken out by the immune system, and then that eradicates HPV from our body. It's after it's in one cell, it's passed on to all of the surrounding cells. Okay. Within the nucleus, it can cause an increase in what we call oncogenes, genes that can cause cancer. We have E6, E7 here. And this causes hyperproliferation of the cells, meaning the cell starts a cell cycle. It tells the cell to start dividing, the human cell, and it will continue to divide over and over. And that's what can cause this atypical growth of these cells, leading to cancer, which can be deadly. Let's go ahead and look at influenza. So similarly, we're going to walk through the steps. We can start with attachment. Here we have our HA attachment. That's our envelope protein that works with the receptor here, tricks the cell to engulf it. Here we see endocytosis. It's being taken in. Here again, it's in an endosome. From that, we're going to go ahead and release the genetic information. Now, this is a negative strand RNA, so a little bit different than what we saw here. So here we were looking at double-stranded DNA. Influenza, on the other hand, we're looking at negative strand RNA. This RNA makes its way into the nucleus itself of the cell. Once we're in the nucleus, we have positive and negative strands here. So we're going to need to go ahead and take the RNA itself, and we're going to start making proteins out of it. So we take some of the RNA and we run the process of transcription, and then we run translation and we start making these proteins. So this is part of the endomembrane system. The virus is hijacking not only our ribosomes. So we have RNA. So we're just doing translation, excuse me. We're doing translation with ribosomes, not only hijacking our ribosomes, but also our endomembrane system in how we develop our proteins. From there, we can also see that we're using our genetic material over here to replicate it, to make more and more of that genetic material. So we go from a negative strand to a positive strand, which then becomes the template to make more negative strands. So we generate our proteins over here. We have these spike proteins that end up on the outside of the cell, and we start assembling our virus. So everything comes back together. We have the genetic material, the negative strand RNA, we have the proteins that it needs, and these viral spike proteins so that it can interact with the other cells. And we see that it's going to go ahead and bud out of the cell. So it's going to take part of the membrane with it. By taking part of our membrane, it's kind of like a camouflage. The virus takes a little bit of our cell as well. And its own spike proteins. What's unique about medications that can prevent the flu, we have Tamiflu, which normally prevents or decreases the severity from influenza. What it actually does is it cross-links the virus against the membrane. Cross-linking it means it's essentially stuck. So it builds up this kind of like a glue assembly and prevents the virus from budding out. So this viral infection stops here. It cannot keep releasing viruses into the surrounding environment to affect other cells. And here we have neuraminidase here, which is going to cleave the host cell receptor, releasing the virus bud out. So that's where our Tamiflu is directly blocking here, that neuraminidase, the NA. Okay, let's move on to HIV. Looking at HIV, this is what we call a retrovirus. So we're going to go from RNA to DNA itself. Looking at the virus itself, it has some unique enzymes. We have that reverse transcriptase, it's called RT. Other things I want to point out are integrase, IN here. Integrase allows us to incorporate the genetic material of the virus into the host cell. So the host cell targets would be human cells like our cells. And so here we see the RNA. It's a double-stranded kind of RNA. It's crossed around itself. So we have one copy of RNA here, one copy of RNA here. So two strands of RNA. Also has a capsid. It's conical, icosahedral shaped. We see spike proteins as well, which can be visualized in microscopy. And again, reverse transcriptase is a really important part, and so is integrase of the virus. Okay, looking at the HIV replication cycle, here we can see our CD4 receptors and CCR5. CD4s are the main ones that are going to interact with the virus. Again, the virus is tricking the host cell to absorb the virus. So here we see the virus is now taken in and it's uncoded within the cell. So here we see the genetic material. And what happens here is that we have a reverse transcriptase in here. So we're going to take our RNA and we're going to make DNA out of it because reverse transcription is going from DNA to RNA. So we're going from RNA back to DNA. And our host, our cells, are made out of DNA. So here we see the DNA moving into the genome, double-stranded DNA. And what can happen here is that it can get incorporated into our genome. Once HIV again is incorporated into our genome, just like any other retrovirus that incorporates itself into a genome, it cannot be taken out. Every time the cell divides, it doesn't only replicate the host DNA, it replicates the viral DNA as well. From here, we have the integrase integrating it in. We need to go ahead and make the viral protein. So here we're going to make RNA. So we go from our DNA to RNA. So that's our transcription. And then from our RNA, we go ahead and make our protein. So here we have our ribosome for the process of translation, generating proteins, again, taking advantage of the endomembrane system that we have here, our complex eukaryotic cells, generating those proteins for the cell membrane, adding them to the membrane. So it transports them to the membrane. And then over here, we have to regenerate the genetic material. We have to regenerate that double RNA that we see. So those two RNA pieces. So we've made our proteins, we made our RNA, and the virus comes back together and buds out of the cell. Now, I like this image because it shows us kind of where we have our antiviral medication here. So a couple of them, if we look at all these steps in the process, again, we have to target things that are not going to harm our cells. And that's what's so difficult about antiviral medicine. So here we see we could try blocking the virus from our, we can block the virus from interacting with our own receptors here. A couple different things. We can block also the virus from being taken in, engulfed by the cell, essentially. So block these signals here. The other thing, so this is preventing the virus from ever getting in. So these antivirals taken here would prevent HIV from ever mounting an infection. So this is what a lot of the medications that people have today that prevent the virus from getting into the cell. But if we have an HIV infection, we could take a mix of things here. So the other ones are suppressing the DNA. We want to keep that DNA within the genome. We don't want it expressing into viruses because those viruses go on to damage host tissue, which we'll see coming up. So a couple other things. We have a protease inhibitor. So protease is going to prevent the production of the proteins that we see here. And that prevents the virus from making that reverse transcriptase enzyme. So by blocking that, the virus, if it doesn't have reverse transcriptase, would not be able to incorporate itself into the genome because it would only be stuck with RNA. So we have a couple different mechanisms, and really what we have today for HIV with the PrEP medications, we have really changed the progression of viral evolution. So 30 years ago or so, an infection with HIV was most likely to become fatal if left untreated. Now, with the antiviral medications that we have today and the preventative methods, it's really changed the course of the course of treatment and the course of the infection itself. So the medications that we have today have overcome a lot of barriers, and we don't see HIV infection now as a direct pathway to fatality because we can suppress the virus from replicating within the body so that it doesn't harm the tissues around it. So now an HIV infection is treated more like a chronic disease, like diabetes that needs consistent treatment. Without treatment, yes, it can become fatal, but with the proper treatment, it doesn't necessarily always amount in fatalities because the virus is not becoming expressed. Also, the medications we have today for HIV treatment, if they suppress the virus within the genome, and the virus never ends up within the blood. So a person could take a test who has an HIV infection, but who is on treatment and be HIV negative from the test. And not only on this assay, on this test, would they be HIV negative, they would also not be able to pass on the virus to anyone via blood or through sexual interaction. Because they don't actively express the virus, there's no way for it to be transmissible. So many people who have HIV infections, who have treatment, cannot even spread the disease while keeping on the treatment itself. It's essentially undetectable, the virus. And that's how good the medication is today that we have. Here's a good list of the antiviral drugs, and this list is ever growing as well. So these are their targets, the mode of action. So we talked about a couple of these, looking at reverse transcription, preventing fusion from the virus ever incorporating itself into cells. And another interesting thing to discuss is genetic modification. We've talked about viruses as a mean of genetically modifying cells because viruses can come in and alter the genome, like we see here. So genetic modification, HIV viruses can actually be used. We can remove the virulent DNA, the dangerous DNA, and use them as a virus to genetically modify human cells because they are the perfect vectors to carry genes into cells. So it's great for genetic modification. Actually, herpes simplex virus as well is great at this as well, depending on what kind of cell we want to target. And then beyond that, we could even talk about mechanisms of genetic modification. So with in vitro fertilization, there was some case studies, experiments done. Actually, a doctor in China genetically modified through IVF the genome of two twin girls. And what he did is he removed the CD4 receptor that wasn't really necessary. He removed a receptor that HIV needs to get into the cell that's not necessary for the cells to function. So by genetically modifying the genes that produce these receptors, he has now genetically modified the individual to be immune to HIV. So if these two young women were exposed to HIV, it would not be able to interact with their cells because they wouldn't have the right cell surface protein. This experiment was very controversial as it was genetically modifying children without their permission or consent, essentially. Noted as illegal, but also a breakthrough in genetic modification. So a lot of morals and ethics that come along with that study. Okay. Let's look at HIV and how it can cause harm in the body. So here on this graph, we're going to see a couple different things. So this is time on the x-axis, and on the y-axis, we have two different things to look at. Here we see our T cell count, and over here we see our HIV RNA copies per milliliter. So we're looking at actually the RNA within the body. So in brown right here we see the virus coming into the body, replicating in cells, increasing. At the same time, we see the T cells dropping, and we see the peak of replication, and we see also one peak here of where the T cells drop. At this point, this is acute HIV syndrome, is what we're looking at here. And we have seeding of the lymphoid organ with the virus itself. So again, it's incorporated within the genetic material. The way HIV works, so it's not within weeks that we see something. People may experience a small degree of not feeling well, like maybe they they feel a little sick. But following that, when we look at the years, is really where we see the impact of the virus. So we can have this clinical latency where, again, the virus is not really actively being expressed. So the patient is relatively healthy. But what can happen is that their T cells will start to drop as the virus will come back. So here we see the virus replicating. This is without treatment, of course. We see weight loss and fatigue. Once someone's immune system is decreased like this with their T cells, we have opportunistic diseases coming in. So anybody who is immunocompromised is more susceptible to disease. So HIV is not actually causing fatality. It's actually an opportunistic disease. So something like gastrointestinal upset, which the immune system can no longer handle, or some kind of cold or flu that the immune system can no longer handle, is where we see our opportunistic disease. Even the own microbiome wouldn't be able to control it, which then leads to fatality. So again, treatment with the medication that we have today changes the outcome, and we don't think of HIV the same way as what we thought of 30 years ago. Now we think of it as a chronic disease, just like diabetes, that will consistently need treatment throughout the lifetime and monitoring. Okay. Looking at retroviruses in general, we can start looking at the human genome. We know that this DNA from a virus can incorporate itself into our genome. But what we don't really think about is the accumulation of viruses within our genome, right? So we can have multiple segments of DNA from viruses within our genome. And if these get into our cells that we pass down to our offspring, then not only is it the viruses that we've come into contact with during our life that get incorporated into our genome, but it's the ones from our parents and their parents. And here we go back thousands and millions of years. And we can look at the evolution of humans. And this is really where we can see viruses changing the trajectory, the projection of humans, and changing the evolution of humans, because viral DNA is accumulating within us here. Here it says half of the sequence of the human genome appears to be originated from viruses and retro elements. I think viruses are a little bit less than 10, so maybe that other 40% is retro elements, those jumping genes that move around that are not really native to our genome. So a lot of these retro elements that we have are essentially accumulations of viruses that no longer can even actually become viruses again. So like looking at this example of bacterial cells and bacteriophage and looking at the options of the lytic or lysogenic cycle. So again, this is what we're talking about, that DNA that's in our genome. We get this accumulation of viral DNA that's passed down from generation to generation as it's copied, just like our own genome. Now, a lot of times once it's copied, mutations are inserted in it and loses the ability to pop back out. So now we just have this old sequence of DNA that used to code for a virus, but can no longer code for a virus because it can't get back into that lytic phase. So it's always just within our DNA copied again. Okay. We've got some interesting facts again about how viruses are really instrumental for us. And again, viruses are not all bad. They're viruses that have shaped our evolution in a beneficial way for survival. And we'll probably learn more and more about that in the future. Okay, we've hit the question point of the lecture. Here we see both HPV, the human papillomavirus, and HIV can remain in a latent state for months to years. In this case, the term clinical latency refers to. We'll go ahead and have you read over the answers. And the answer is a period of time after the initial infection when the virus is not causing symptoms and is virtually undetectable. So that's our clinical latency. So again, with an HIV test, especially with the treatments we have today, it's virtually undetectable in the system because we don't have active viral replication and it cannot be passed on to people. HPV, again, can be the same way. So it can incorporate itself within the genome and not be actively expressing. Question number 2. Which of the following is not a reason why antiviral agents are hard to develop? Go ahead and take a minute, pause the video to read through the answers. And the answer to question number 2 is to destroy the virus, the host cell must also be destroyed. So when it says which of the following is not a reason why antiviral agents are hard to develop, let's go ahead and read through them then, because these questions essentially tell us that the rest of the answers are correct. So the answer number 1 is because the viral genomes are small, there's relatively few molecules that can be used as drug targets. So less DNA, less drug targets, less proteins that it makes. Antiviral agents usually have severe side effects because viral replication involves many of the host cell processes. Again, yeah, we're using the DNA polymerase, the RNA polymerase, we're using the Golgi apparatus, the endomembrane system. So we can't target those because that would harm our own cells because that's our own cell machinery and metabolic activities. Okay, then the last one here is drug-resistant strains develop rapidly because viruses mutate quickly within the host. Yeah, they mutate quickly because they have that small genome again, so they can actively bring in mutations and adapt to new environments. So just as we see antibiotic resistance, we will see antiviral resistance as well. Okay, so question number 3, which of the following viruses is dependent upon an RNA-dependent RNA polymerase for replication? We'll go ahead and look at the answer for this one. So we're looking at dependent upon an RNA-dependent RNA polymerase for replication. So this is where you can extrapolate going back to the Baltimore model. So here we see influenza is a single-stranded RNA virus. So an RNA-dependent RNA polymerase is actually an enzyme that takes RNA and makes RNA out of it. So a single-stranded RNA strand, we can go from the sense to the template strand or coding to non-coding strand. So that's a really, really unique enzyme. Again, we don't have an RNA-dependent RNA polymerase. This is a unique enzyme that's developed in viruses. Again, viruses can infect all forms of life. They're the most ubiquitous. So we see all of this diversity within viruses as well. So influenza virus is a single-stranded RNA virus that would need an RNA-dependent RNA polymerase in order for it to replicate. Okay, question number 4. Hypothesize which characteristic of the influenza virus makes the genome particularly susceptible to antigenetic drift. Okay, the answer to question number 4 is the genome is segmented. So when we're looking at why it would be more susceptible to antigenetic drift, the segmentation allows for that movement more. And allows for that diversity, that change. Okay, we've made it all the way to the section objectives. Again, to read these over and make sure that you're getting the main points of the lecture. Sometimes listening without following along with the book or engaging with the material gives us a false sense of understanding the material. So going through section objectives is making sure that you're hitting the critical points. And these are your study guides. So you do a study guide after each lecture instead of waiting to do a comprehensive study guide of multiple sections. Okay, and that is the end of the virology lecture.